Assalamu alaikum. So let's continue talking about regulation of transcription. Right now we'll be talking about regulation of transcription in eukaryotes. So the thing is, things are more complex in eukaryotic cells. So there are more elements, more proteins, more genes, and so on. So everything is more complex, except that we're talking about the same principles, okay? The same basic principles. That is, you have non-covalent interactions, you have activators, you have inhibitors, you have DNA binding sites, elements specific, that is specific for certain proteins, okay? Now, the thing with regulation in eukaryotic cells is that there is more control, higher levels, or more levels of control. So we have cis-acting elements, just like in bacteria. There are promoters, promoter proximal elements, enhancers, silencers, and so on. There are transacting factors, proteins, and these proteins can be transcriptional regulatory proteins, activators, repressors, and what they do is that they can modify chromatin and DNA specifically chemically. Okay, so they can modify histones, and they can also modify the DNA itself, the structure of DNA, the nucleosomes, in other words. And DNA itself can be modified as well, the nucleotide, specifically cytosine, by methylation. Now, we also have something that bacteria do not have, as far as we know, and that is non-coding RNA molecules. There are many non-coding RNA molecules that play a role in regulating transcription. So we'll talk about all these levels of regulation in eukaryotic cells. So what we have in here is the same concept again. We can have different types of activators. You can have repressor as well, and there are elements on the DNA. Okay, so you can have activators binding to these different elements, depending on the activators, depending on the types of activators, and depending on the number of these activators. You can play around with tuning up the level of transcription. So transcription can be very effective. That is really strong transcriptional activity, or you can have very little or hardly any transcription whatsoever, depending on what binds to these elements in the DNA sequence. Of course, if you have a repressor binding to DNA, you can have inhibition or block of transcription altogether. Okay, so same concept again. So how do transcription factors regulate gene expression? Now what happens here is that what they do is what we call epigenetic changes in DNA and chromatin. So if I say that there is a genetic control, or if I say there is a genetic change, what we're talking about is a change in the sequence of DNA, in the nucleotide. So we change a cytosine to guanine. Okay, that's a genetic change. But if I say epigenetic, or if I say epigenomic, what we're talking about here is we're changing the structure of DNA and the structure of the chromatin itself. So this is a higher level of regulation, and that's what epi means. So epi means above or in addition to. So in addition to the genetic change, we have something even higher, and that is the epigenetics. So what it indicates is genetic alterations in gene expression without a change in the DNA sequence. And here what we're talking about is basically changing the packaging of DNA, the chromatin structure, chemically modifying histones, and chemical modification of DNA. Again, same as in the previous slide. So let's talk about transcription factors and how they look like. So basically, these transcription factors have at least two domains, and we're talking about activators. These domains are DNA binding domains, or DNA binding domain, and an activation domain. So what do we mean by a domain? So a domain is basically a three-dimensional structure. So it is part of a protein. Okay, so you can imagine a protein having, like, let's say, two arms or two hands. Okay, you have a DNA binding domain and you have an activation domain. Now, so it's basically a certain structure. Okay, so and they look different, of course. Now, this three-dimensional structure, the DNA binding domain binds to DNA, and the activation domain would do whatever action it does. Okay, so usually these domains are independent of each other. So meaning that if you separate these domains, each arm or each hand can function by itself independent of the other hand. Okay, so that's the idea of a domain biochemically. So they can be separated. Now, the activation domain is the part of the protein that activates or stimulates transcription. And it does so by interacting with general transcription factors. So here you have the DNA binding domain, okay, and you have the activation domain. So the idea is that you can cleave it like here, for example, and this would still be functional. And this can be functional as well. Okay, so this part, the activation domain, what it does is that it stimulates transcription by interacting with other proteins. Now, these other proteins can be general transcription factors, specific transcription factors, mediator, which binds to the enhancer, DNA modifiers, and so on. Okay, now, or what the activation domain can do is that it can modify the structure of the chromatin. Okay, and we'll talk about that in 5 minutes or so. So how about repressors? I mean, we talked about activators, okay? How about repressors? Now, these repressors, there are two types, and you can either have a repressor having both also a DNA binding domain and a repressor domain. Okay, so what it does basically is that it has both a DNA binding domain right here, and it has a repressor domain, and this domain, this structure right here, would bind to the general transcription factors, the mediator, RNA polymerase, whatever, pre-initiation complex, any other proteins, and what they do is that they block transcription. Or these repressors can only have one single domain, which is the DNA binding domain. And it binds to the DNA, and it prevents the activator from binding to the DNA. So there's competition between the repressor and the activator in binding to DNA. Okay, so you have this activator binding to the DNA. Okay, to a DNA element, to the promoter proximal element, whatever. Now, what they do, or one thing they do, is that they can modify the chromosomal structure. So what do we mean by that? Remember, we talked about two types of structures, and we said you have what is known as the heterochromatin. This is where the DNA is highly packed, and there is no access for proteins to bind to DNA because all of the DNA sequences are hidden, are wrapped around the histone core. Okay, now, or you can have the euchromatin. Here you have a loose DNA. You have the DNA that is accessible to transcription factors, inducers, stimulators, activators, you know, all of these names. Okay, now actively transcribed genes exist right here in euchromatin, and packed and inactive genes exist in DNA that is a heterochromatin. Okay, so remember when I said that all cells have the same DNA. Okay, they have the same genes, but they express genes differently. And one mechanism or one reason why they express genes differently is that they produce different transcription factors. Okay, and the other reason is that the DNA is packed differently in these different cells. So you have in one cell you have a gene in a euchromatin, in another cell you can have the same gene, but it exists in a heterochromatin. Okay, so what regulatory, even within the same cell, by the way, what transcription factors do, or these regulatory proteins, is that they can switch genes between both structures, euchromatin and heterochromatin. So whenever cells want to activate a certain gene, they loosen the DNA so that they make DNA accessible to transcription factors. And whenever they want to silence genes and block transcription, what they do is that they pack the DNA where the gene exists so that it cannot be accessed by transcription factors. Okay, so how does this happen? This packing, unpacking of DNA? Well, one mechanism is via chromatin remodeling factors. Well, from that name, you can tell what they do exactly. They remodel DNA. They change the shape of DNA, the structure of DNA. Okay, and they do so by one of three mechanisms. They can remove histones altogether from DNA, so that now DNA is accessible. It's free. Okay, they can reposition nucleosomes, making DNA accessible again. So what they do is that you have a remodeling factors, and what it does is that it binds to DNA and it pushes the histone a bit further. Okay, so what it does is that it frees a certain DNA sequence, like a terabox, that a transcription factor 2D binding site and the RNA polymerase 2 and so on. Okay, or what they do is that they alter nucleosome structure, okay, by allowing proteins to bind to DNA. Okay, so it's different than repositioning nucleosomes. What they do again is that they change the shape of the DNA, again exposing a certain sequence to regulatory or transcriptional proteins. Now, these, by the way, chromatin remodeling factors, they are associated with activators and repressors. Okay, so activators can do this, right? They can make sequences accessible, free of histones and so on. Repressors do the opposite, right? So what they do is that they hide these sequences. They make the DNA highly packed. Now, one mechanism of changing the structure of nucleosomes or switching DNA from the euchromatin structure to the heterochromatin structure is by modifying histone 1. Okay, that is allowing it to bind or preventing it from binding to DNA. So there are certain proteins, regulatory proteins, wherever the names of these proteins are, what they do is that they allow histone 1 to bind to DNA, and what histone 1 does is that it really packs DNA. Or these proteins can remove histone 1, again freeing DNA sequences. How can chromosome structures be altered as well? Well, there are two mechanisms. So in addition to remodeling chromatin, there are two mechanisms, and these mechanisms include chemical modification of histones, and the other mechanism is binding of non-coding RNA to DNA. Okay, so there are two mechanisms that can switch DNA from heterochromatin to euchromatin and vice versa. Vice versa means opposite is true. So let's talk about one particular mechanism of of chemical modification of histones, and that is histone acetylation. So what do we mean by acetylation? This is an acetyl group right here. Acetylation means addition of this acetyl group to another group. So you have this, so the OH is replaced by another group. Okay. Now, if you look at histones carefully, well, not you, but when scientists looked at histones closely, they found out that histones have two domains. Again, what do we mean by domains? Three-dimensional structures that are part of a protein, okay, and they are internal. Now, there are two domains, and these domains, one of them is known as a histone fold. So the way it's like folding clothes, okay, or t-shirts, whatever. Same thing with proteins. They can be folded in certain ways so that they have a certain three-dimensional structure. So it's called one domain is known as a histone fold. Now, histone fold is basically the internal structure of histones, and that's where histones interact with each other and interact with DNA as well, wrapping DNA around it. Okay, so the histone fold facilitates the formation of the nucleosome core particle. Now, the other part, the other domain of histones is basically a tail. Okay, and this tail exists at the amino terminus of these proteins. Now, this tail right here is rich with lysines. So what is a lysine? It is an amino acid that constitutes, that makes up the histone molecule. Okay, so what is lysine? This is the structure of lysine. Okay, so it is an amino acid, and it is a positively charged amino acid. So it has this positive charge right here. Now, this positive charge facilitates the interaction between histones and DNA, because DNA is negatively charged, right? Well, the thing is that when histones have lysine and whenever they have lysine with the positive charge, now the interaction between histones and DNA is very tight, okay? Because there are many histones, and again, there are many lysines in histones. Now, what happens is that the lysine residues, the lysines in the histone molecule can be acetylated. Okay, so by an enzyme known as acetyltransferase. So it's known as histone acetyltransferase. Now, when lysine is acetylated, the positive charge is masked. It's removed. It gets removed, right? So the interaction between histones and DNA becomes weaker, and DNA becomes loose because of this weak interaction between histones and DNA. So actually, what activators and repressors have is that the way they function is that they get associated with histone acetyltransferases that transfer the acetyl groups to lysines, masking the positive charge, or they can be associated with deacetylases. These are the enzymes that remove acetyl groups from lysines. Okay, so again, what histone acetyltransferases do is that they transfer acetyl groups to histones, weakening the interaction between histones and DNA, allowing for the formation of Euchromatin, okay, and facilitating or activating transcription. On the other hand, what deacetylases do is that they are inhibitors. What they do is that they remove the acetyl groups from lysines. So now you have the positive charges now clear, apparent, obvious to DNA. You can have this strong interaction between histones and DNA becomes highly packed. Okay, and it turns out that transcription factor 2D specifically has a histone acetyl transferase. Okay, so activity. So what it does is that it's the first protein that binds to the promoter region. It modifies DNA so that it becomes loose, accessible to other proteins, allowing other proteins to bind to different sites on DNA and inducing transcription. Now, there are other modifications of histones. So they can be methylated, they can be phosphorylated, and the effect is really dependent on the site of modification, that is, to which lysine are these groups added. Okay, and it's a bit sort of complex, okay, because there are many lysine residues. These are the ones represented as Ks. So there are many lysine amino acids that make up this histone molecules. Depending on which lysine is modified, you can have activation of, in general, you can have activation, induction of transcription, or repression of transcription. But overall, what acetylation does is that it represses transcription. And just like the DNA polymerase has a problem replicating DNA, reading DNA in the presence of histones and the histone core complex and the presence of nucleosomes, the RNA polymerase also has the same problem. So histones must be removed from DNA in order for the RNA polymerase to transcribe the DNA and synthesize RNA. And this is done using elongation factors. Okay, now these elongation factors associate with the RNA polymerase, and they specifically bind to the tail of the RNA polymerase. Remember this tail that gets phosphorylated, and then you have the capping factors, splicing factors, polyadenylation factors. Same thing with elongation factors. They bind to the phosphorylated tail of the RNA polymerase 2, and as soon as RNA synthesis is initiated. Okay, and these elongation factors, what they do is that they remove histones. They have these histone modifying enzymes, they have the histone acetylases, or the acetyltransferases. So what they do is that they acetylate histones so that their interaction with DNA becomes weaker. And they also contain chromatin remodeling factors that remove histones and dismantle nucleosomal structures. Now, we also have a role for non-coding RNA molecules. There are, specifically we're talking about long non-coding RNAs. Now these are long because, well, relatively long to differentiate them from smaller RNA molecules, also known as microRNA, as well as other types of RNA like circular RNA, PV-RNA, and so on. There are over 50,000 long non-coding RNAs. These have been discovered in the past 5 years or so. And you just imagine the immense amount of research that is needed to identify the functions of these long non-coding RNA molecules. So you can just imagine, we're not talking about 50,000 different research articles that can be published. We're talking about like over 1 million research articles that can be published, or research work that can be done on these RNA molecules. Now these long non-coding RNAs can be homologous to certain DNA sequences. So what they do is that they can bind, they can let's say hybridize, they can anneal to certain sequences of DNA. Of course, once they do that, many things can happen. Among them is that they can associate with different proteins and different enzymes that can modify the chromatin. They can modify, they can chemically modify the histone molecules via, for example, methylation. They can regulate chromatin condensation and packaging. Okay, that is relaxing or packaging of DNA molecules. Now these long non-coding RNA molecules can also bind to general or specialized transcription factors such as the general transcription factor 2B. They can also bind to RNA processing proteins. So, again, we don't know much about what they do, but they do a lot of things. What's really fascinating as well is that enhancers amazingly can be transcribed into RNA molecules, and these RNA molecules are also known as eRNA. E stands for enhancer. Okay, now what these eRNA molecules do is, again, we don't know exactly what they do, but it appears that they influence transcription of nearby genes. Okay, again, we don't know exactly what they do. Let me talk about one type of RNA, of long non-coding RNA molecules, and this molecule is known as ZIST. Now, by the way, these long non-coding RNA molecules can act in cis or trans. What do we mean by that? Just like e-enhancer eRNA can influence nearby genes in a cis manner. Okay, cis means same level. You can also have the production of long non-coding RNA molecules that can travel to other chromosomes and influencing transcription of other genes on other chromosomes. Okay, so they can act in trans as well. So I'll talk about one type of long non-coding RNA known as ZIST, and ZIST acts in a cis manner. So here's the thing. Now, females have 2 X chromosomes. Males have 1 X chromosome. This is not fair, of course, okay, because females have more. So what happens, and it's pathological as well to have 2 active X chromosomes, okay, double the amount of genes. So what happens in females is that randomly one of the X chromosomes in cells gets inactivated. So this is called a phenomenon called dosage compensation. So the idea here is that at the end you have 1 active X chromosome in females and 1 active X chromosome in males. Now in females it's a combination. That is, inactivation is random. So you have some cells having 1 X chromosome active, and in other cells you have the other X chromosome active. Okay, so it's called mosaicism. Okay, so here's the thing. So the inactivation of this X chromosome occurs this way. The first thing that happens is that you have transcription of the ZIST RNA from the X chromosome to be inactivated. Okay, when this long non-coding RNA is produced, what it does is that it coats the X chromosome that produces it. Now this promotes the recruitment of a number of proteins that methylate histone 3, and that leads to chromosomal condensation. Okay, so the chromosome gets shrunk. It shrinks literally, and it becomes what is known as a bar body. So in a female cell, or if you look at a nucleus, you would have the whole chromatin right here, and you have the inactivated X chromosome isolated and shrunk, by itself. Okay, so again, this is known as dosage compensation. So what happens is that you have one active X chromosome in males and one active X chromosome in females. So again, you have transcription of a long non-coding RNA known as ZIST. This RNA molecule codes the X chromosome to be inactivated. This is followed by methylation of the histones, and that results in condensation of the X chromosome. Of course, you have other modifications as well, such as hypoacetylation, that is removal of the acetyl group so that the interaction becomes really strong between histones and DNA is packaged tightly. Now, here's another modification. And this occurs not to histones but to DNA itself. Now what happens is that some of the cytosines can be methylated, producing what is known as 5-methylcytosine. Now please pay attention to the structure of this methylcytosine. Go back to lecture 1 and look at the difference between the structure of methylcytosine and uracil. Now what happens is that some genes, the promoters of some genes, can have a CG rich sequence. So a lot of CGs in the promoters, and this is called CPG islands. What happens is that the cytosines in these CPG islands get methylated. Methylation of cytosine results in reduction of transcription, in other words, inactivation of genes. Now, it's really common in cancer cells that you have disturbance in cytosine methylation, leading to activation and inactivation of certain genes. Now, DNA methylation is important because it is related to a phenomenon known as genetic imprinting. So what do we mean by that? See, the thing is that there are certain genes that must be transcribed specifically. That is, in other words, it's either the maternal gene that has to be transcribed or the paternal gene. Okay, or if you have or both genes must be activated. If you have inactivation of one of these genes, if you have production of only the maternal gene or the paternal gene, certain diseases can result from that. So this happens, this is known as genetic imprinting, that is controlling maternal and paternal gene expression via, and this is done via methylation. So epigenetics is a really important phenomenon in regulating gene expression. It's very complex. We are still trying to understand how it works and what it does exactly. And yes, you can have, by the way, twins having the same exact DNA sequence. Yet, if you look at their DNA and their genome, you can find different pattern of methylation and acetylation of histone molecules and DNA as well. And that results in differences in gene expression. Okay, so that's why they look different. Somehow there are differences between them. So, one of them can lead a healthy lifestyle, taking vitamins, playing sports, and the other person could be a menace, leading a really unhealthy lifestyle, drinking carbonated drinks, Pepsi, Coca-Cola, and whatever, and eating a lot of candy and chocolate and etc. That leads to this person having a different genome, or I should say epigenome. Yes, Again, twins can have the same exact DNA sequence, but they have different epigenome, and that leads to subtle differences in their looks, maybe their behavior as well. Okay, so these mice, they have different colors, even though they have the same genome, the same DNA sequence, as a result of having a different epigenome. So the thing is that having a certain DNA sequence does not mean that you will have a certain phenotype. Okay, so you can control your own destiny, by the way, and do not blame your failures on your DNA. Okay, so you can control that by controlling your epigenome, your DNA modification. And this is an interesting article that that shows that stress can change DNA modification, and as a result, DNA expression. And by the way, DNA modification and epigenetics can be inherited as well. Okay, so the CDC, that is the Centers for Disease Control in the US, um, um, says that okay, so let's wrap up everything. Okay, we're gonna put things together and talk about a scenario of how things work inside the cell. So the first thing that happens is that you have chromatin remodeling factors, you have all of the other enzymes that bind to the DNA and chemically modify the chromatin or structurally modify the chromatin. Okay, so what happens is that the important sequences, like the Tata box, for example, they are hidden. And these remodeling factors and proteins, what they do is that they modify the DNA so that these sequences get exposed. Now, the sequence here, this one right here, uh, represent this whole gene right here that includes the promoter proximal elements, exons, introns, and so on. So this is step number one. So right here you have exposure of the promoter. So it's not packed anymore. It is exposed. It can be accessed by the pre-initiation complex. So what happens now is that you have the formation of the pre-initiation complex on the promoter. Okay, so following exposure of the promoter, you have assembly of the pre-initiation complex, which can also interact with the other regulatory proteins that are bound to the promoter proximal element, as well as the enhancers. Okay, then what happens afterwards is you have attachment of the RNA polymerase, and it starts to transcribe genes. Okay, so let me give you an example of a transcription factor, and this is a fascinating, a very nice, well-studied example um, involving nuclear steroid receptors. So if we look at the structure of these nuclear receptors, they look like this. Okay, sort of. Okay, so they have three domains. Okay, again, we talked about domains. These are internal three-dimensional structures, and they function independently of each other, so we can represent them as such. So right here you have a ligand binding domain. Now, a ligand is basically a small molecule, like a hormone, for example. So I have a hormone that gets into the cells and it binds to the ligand binding domain. Then you have a DNA binding domain. So this is the part of the protein that binds to the DNA. And then you have an activation domain. So you have a third domain. This is the domain, this is the part of the protein that can bind to co-activators or co-repressors. This is the part of the protein that would activate transcription or repress transcription of certain genes. Okay, so this is how a steroid nuclear receptor look like, basic structure. So this is how it functions. This is how these receptors work. You have a steroid. A steroid involves, or it includes, things like hormones such as androgens, the male hormone, sex hormones, estrogens, the female sex hormones, progesterones, again the female sex hormone. You have cortisol or cortisone. It's a very important hormone that regulates a lot of functions inside our body. And then you have aldosterone as well. It's another steroid hormone. Okay, aldosterone can regulate kidney function. It regulates the amount of water inside our body. Now, so these hormones are small and they are hydrophobic, meaning that they are lipid-like. So they can diffuse through the plasma membrane inside the cell. Once they are inside the cell, they bind to their receptor. Okay, this receptor right here. So the ligand, the hormone, binds to the ligand binding domain. What happens then is that the receptor dimerizes, meaning that it forms a dimer with another receptor. So you have two of these receptors right here. Okay, this one right here. It binds to the hormone, and two of them can dimerize. So they form a dimer. They form a complex together. Then this dimer gets inside the nucleus. So right here you have the nucleus. The dimer gets inside the nucleus, and the dimer can then bind to a hormone response element. Again, an element is what? It is a specific sequence in the DNA. And this element, again, it's a specific sequence that binds to this domain right here. Now, once the receptor that has the ligand bound to it binds to the hormone response element, it recruits many proteins. Now, these may include coactivators. Now, what happens then is that these coactivators, so the hormone response element, by the way, is a promoter proximal element. It's close to the core promoter. So interaction takes place between these proteins, the complex right here, with the pre-initiation complex that binds to the promoter. This interaction activates the RNA polymerase II to start transcription. Okay, now, so the activation domain right here is the one that is responsible for interacting and binding to all of these proteins, or in forming the whole protein complex. Now, once you have this happening, this interaction, then you have this interaction with the pre-initiation complex activating RNA polymerase II to start transcription. Now, same thing with other receptors, okay? The cell surface receptors, that is. So you have these cell surface receptors, and each one of them can bind to its own ligand, okay? Hormone, growth factor, you know, what have you. So what happens is that you have activation of this receptor once the ligand binds to it, and you have signal transduction activated. And that results in, let's say, activating of one protein, which activates another protein, which activates a third protein, and so on. So that's signal transduction. Eventually, the signal is relayed to inside the nucleus, and you have activation of co-repressors or co-activators, and they bind again to promoter proximal elements, enhancers, and so on, changing the structure of the chromatin, either loosening it so that these co-activators and repressors and regulatory proteins can bind to regulatory elements, or compacting it, packing the DNA so that it cannot be accessed by all of these proteins. Okay, so this is how things work inside the cell. So again, you can have either activators binding to the DNA, promoter proximal elements, enhancers, whatever, activating transcription, or you can have repressors again, influencing gene expression, blocking transcription, inactivating gene expression, and so on. Okay, so this is how genes are regulated in eukaryotic cells, and there's more to come, of course, because there's always research going on. Allah ma'akum.